We've been using and exploiting sort of genetic and DNA-based information for close to 20 years from all sorts of organisms, from viruses to bacteria to fish to humans, in order to accomplish very specific tasks and troubles and challenges that various industries and individuals and companies face. So it's been a lot of fun. I've only been there for 6 years, but it's a great place to work, a lot of fun. Just a little bit about RPC, I guess. Been around since about 1962, acting as New Brunswick's provincial research organization. We have a staff of about 98 scientists, engineers, technologists, and support staff. We serve about 800 clients annually. 2/3 of those are from New Brunswick. The other third comes from the rest of Canada and probably about 30 different countries worldwide. So we tackle a lot of different problems. It's rare that we'll say no to anything. We do work in a lot of different sectors, from energy and manufacturing to natural resources to the healthcare, mining, and aquaculture. So we're very focused on industrial problems, helping them solve issues that helps them move along in their business to be competitive on a global and local basis. I'm head of the Food, Fisheries and Aquaculture Department. We're sort of the experimental and applied bioscience group at RPC. We have a fairly healthy staff of PhDs, MSCs, BSCs, and people with technical degrees from college. So we have a well-rounded group doing a lot of different tasks. What I'm going to talk about is some of the genomics-based areas that we utilize in our everyday work, and it's primarily going to be focused on the aquaculture industry. My department kind of grew up alongside the aquaculture industry as it was developing and growing here in New Brunswick, and so we've been helping that industry a long time with a lot of the issues that have come up. And I'm sort of going to focus on two different areas. One I'm going to focus on is our technical services, things like fish pathogen surveillance and strain typing of pathogens. This work here derives pretty much all of its information from knowledge about the genome from these various pathogens, because we're targeting very specific areas in order to detect and find out if they're present. The other area I'm going to talk about is some of the R&D that we do in my department as well. Things like DNA traceability systems, how we're using genomics to improve vaccines, and just sort of dabble a little bit with some of the new frontiers that we're sort of looking at. All of our projects and other things always usually start with a challenge or a problem, and one of the main challenges, of course, is managing disease. It doesn't matter whether you grow potatoes or you raise pigs or you grow fish. Diseases are always a constant issue that you face when you're doing that kind of work, and outbreaks of disease can cost millions of dollars. There's no question about that. To be preemptive of that, there are various tools and strategies that can be employed, and of course one of them is having effective surveillance tools, nip it in the bud, so to speak, and of course having effective vaccines. Now, these are very preventative strategies, of course, because as the old saying goes, an ounce of prevention is worth a pound of cure. And some of you have probably seen this pyramid before, in that, you know, for every dollar you spend on prevention, you're saving yourself 100 in trying to clean things up if things go off the rails. So there's very good economics behind being preventative in terms of managing disease. Solution, of course, is having good diagnostics, and effective surveillance requires very sensitive and highly specific diagnostic tests. Right? They establish the pathogen identity causing the issue, and that can be viral or bacterial. And basically the detection strategies that we used are based on targeting genetic elements which are highly specific to each individual pathogen. So we are looking for genetic information that is very unique to the pathogen that we are trying to detect and screen for. So we use a variety of technologies. Here's one example. It's a high throughput technology that we use so that we can screen large numbers of animals at a time to provide very rapid and efficient results to our clients in order that they can make decisions about whether their fish are at risk of developing disease. We're also using a lot more sensitive techniques than we were, say, 5 or 10 years ago. The methodologies that we're using now is probably anywhere from 10 to 100 times more sensitive than those older techniques. And so being very specific and highly sensitive is the name of the game in pathogen diagnostics. Complementary to that pathogen diagnostics is also having a capability to understand something about the pathogen that you do find. With some pathogens, specifically viral pathogens, you can have various strains of that virus, some of which may be lethal, some of which may be somewhat benign. So here's an example here. This is genomic information from the actual virus. And this is a database of viruses that we maintain, a fraction of them. And basically, if we detect a virus and we sequence its genome, a portion of its genome that we know is associated with virulence of that pathogen, and we see this type here, generally that's not a great cause for concern. Quite often the pathogen will pass. If left enough time, you keep an eye on your organisms and they'll be quite happy. However, if we happen to detect one of these ones, then that's significant, because that could mean that your animals are facing anywhere from a 60 to a 90% chance of mortality, very high losses. So having a database and having knowledge about virus genomes and which ones are lethal and which ones are not are very important to industry, because it helps them make management decisions about what they need to do with their fish. It also helps government regulators so they can assist and make some decisions about what they need to do in terms of preventative actions. The second sort of preventative solution here is vaccines. Vaccines are very proactive in helping prevent disease, helping organisms adapt to their immune systems so that they can fend off an infection. This is a project here that we're working with Novartis on to improve a vaccine for ISA. And you wonder what this microscope slide has to do with a project such as this. But basically, if you look closely at that microscope slide, you'd actually see that it's imprinted with about 44,000 genes from the Atlantic salmon genome. And this is a very effective tool to try and gauge how a salmon responds to ISA infection and how it responds to a vaccine. Ideally, you want your vaccine to prompt a very similar response in the fish as it does with an actual virus infection. And so by looking how a salmon responds at a genetic level, you just have to trust me that there's about 44,000 spots on this graph here indicating the response of the salmon genome to either a virus infection or to a vaccination. And the idea is that we can see what genes are sort of downregulated and what genes are upregulated in terms of that response. And the idea here is to try and marry the two, whereby we can create a vaccine that shows a response very much identical to what a pathogen would exhibit as well. And so from that list of spots, and each spot represents a different gene, we can develop a list of biomarkers that become a very good indicator as to whether or not your vaccine is going to be efficacious or not. So the benefits of that system, of course, it allows for the identification of vaccine performance markers to mimic the pathogen response in the fish. So you can increase the rate of vaccine development that way. You can increase the efficacy of the vaccine, which is the ultimate goal, of course. And you can decrease your development costs. So there's a lot of benefits to this type of a system. And of course, this all leads to better products and happy customers down the road. Some other challenges. Escaped farm salmon. This could be a challenge. Food safety and consumer confidence is a challenge. Operational sustainability. This is a challenge. Is there a common solution to all of these problems? Well, this is a solution to all of those problems, and it's a DNA traceability system called Offspring that we've been working on with Cook Aquaculture in the University of New Brunswick. This is basically a complete egg-to-plate traceability system, and it addresses each and every one of those components that I mentioned earlier. If we look at the production cycle of fish very generally, there's a parental phase. The parents, they produce eggs. This usually occurs in a hatchery. Then there's the fish hatch, and there's a grow-out period. They go through processing, shipping, and wholesale. Then the fish enter the retail chain and ultimately end up on your plate. And so the various facets of this traceability system cover various aspects here. So in the very beginning, we start off with the system of DNA fingerprinting and databasing all of the parents. So we know the genotypes or the genetic fingerprint for all of the parental fish that go into producing production fish. And we put those in a database. In the next phase of the chain, there is a whole chain tracking software to cover where the fish move, what kind of feed they received, what kind of treatments, other types of things that may have gone on with the fish, where they were processed, where they were shipped. It stores all of that information. And the real outcome happens down in this end here. So if there was an issue with food quality, with a fish fillet, or perhaps a salmon shows up in a stream that is suspicious as to where its origin, we could take a tissue sample from that fish, genotype it, do a database query back to our parental database, and using parental assignment software, we can actually trace the entire life history of where that fish came from. We can see who its parents were, what hatchery it was raised in, what feeds it received, what treatments it received, all of those aspects which is quite a powerful technology. And after visiting around, we've come to know that this is a world-leading technology that we've developed. No one else in the world is doing this level of DNA traceability with a product. So what are the benefits? Well, to the escape issue, they can be monitored and the origin determined. Food safety and consumer confidence, we now have whole food chain traceability. That's a very powerful thing, and in the food production circles, this is the buzzword. Everybody is trying to achieve some whole chain traceability for their products. Where did this ingredient come from? Is it produced organically? Where did that ingredient come from? Is it come from a sustainable stock? All of that information. And having that capability of tracking, you can command premium pricing for that. People will pay extra money to have a product like that. Operational sustainability. Because we're looking at the genetics of these fish, we can minimize inbreeding of the stocks and track the family performance. And the great thing is that this system is deployable across multiple sectors. It doesn't just work with fish. It can work with multiple species of fish. It can work with terrestrial animals. It can work with a lot of things. If it has DNA, we can trace it. The last thing I want to touch on here, I guess, is kind of dabbling in some new frontiers. And our dabbling began in a tide pool filled with scum and slime with a few swabs. Because from this, you can get this. And these are purified strains of marine bacteria with a variety of different properties. So from this, we then developed something like this, where we're combining genomic screening with bioinformatics and assay-based selection. So we're taking information from the genomes from each of these bugs and combining it with a bunch of other information and kind of getting a figure like this, which I'm not going to explain, but it's basically a decision information piece that has come from the result of probably tens of thousands of screening assays to allow us to focus on one particular bug. So this is what we end up from about 2,000 different isolates. We come down to one that has something of interest in it. And that something of interest is a valuable molecule. And in this particular bug, we found it has a compound called violacine, which has a very broad spectrum of activities. It has antibacterial properties, antiviral, anti-cancer, and anti-trypanosomal. It has a very specific target within a cell that allows it to deal with a lot of different things. And this is very exciting and interesting that we have several different strains of marine bacteria that produce this. If you were to buy this compound from a chemical company today, it costs you about $330 per milligram. And if you grow these bugs, you can produce about 1.1 grams of crude compound just in a liter of culture. So in a liter flask about yay big, you can produce about that much. So say only that's 50% pure. Say you have 550 milligrams. Well, and say the company's only going to pay you $150 a milligram for that. Well, from 1 liter of culture, there's about $80,000. Really? That kind of gets exciting considering the price of crack cocaine is about $600 to $1,000 an ounce, and that's 28 grams, right? So it all depends on the market, right? A lot of this compound is currently undergoing a lot of clinical trials, of course, and if those clinical trials are then successful, there's going to be a huge demand for the production of this compound. And of course, marine bugs and the biochemistries that they have, there's no better synthetic machinery than that. Okay, what's the benefit of this? Well, it could create new jobs. There could be people tasked with just growing up and producing this compound. And of course, the effective treatments that result as well. So we work with a lot of different partners and a lot of different companies. You know, like I say, we rarely say no to anything. If it's got an intriguing solution that we can apply genomics to, we're more than curious enough to take it on. So some of the benefits that we've sort of seen from using genomics, you know, this solves real-world problems in a very efficient and effective way. It enables global competitiveness for our clients, allows the development of new capacities. When you change the way you do things, suddenly other things become possible that you couldn't imagine before. It's a source of very highly skilled jobs. Many of the people that work for me, they've been educated right here in New Brunswick. And it's a great thing. I'm from New Brunswick. It's a great thing for me to have an advanced education and to be able to come back here and find employment in my field. And it makes me feel good that I can hire people that have education in that field as well that want to stay here in New Brunswick. But these things require continued support, of course. Some of this research is not cheap. And we need to do this to keep pace with other countries, because other countries definitely see the value in this and are pumping reams and reams of money into research to do this. So to keep our industries competitive with these other countries, we need to keep investment going here. We need to stay ahead of the curve. Thank you.